Good afternoon, and welcome to Season 4 of Itasca Water's Practical Water Wisdom series, which focuses on issues related to water and ways to resolve those issues. I'm your host, John Downing, Director of Minnesota Sea Grant and an Itasca Water's board member. I'm sitting in for your regular host, Brian Whittemore, who's away today. Today's episode is called Harmful Algae Blooms and Climate Change. Itasca Waters is a nonprofit volunteer organization headquartered in Itasca County, Minnesota, for the purpose of educating folks about the value of clean water for the environment and the economy. We offer a ton of good information for full and part-time residents through our programs, and it's all available on our website at itascawaters.org. We thank these partners who have been with us all along for helping Itasca Waters offer our Water Wisdom series. Dr. Chris Philstrip will speak to us for about half an hour today and then take your questions. To submit a brief question, just click on the Q&A button on the bottom of your screen and type it in. You can do that at any time during today's program, and we will read the questions on your behalf. Today's program is being recorded and will soon be available on our website, itascawaters.org. As we mentioned, the subject for today is harmful algae blooms and climate change. And our expert presenter is Dr. Chris Philstrip, who's a limnologist at the University of Minnesota Duluth's Natural Resources Research Institute, an applied research institute that delivers integrated research solutions that value our resources, environment, and economy for a sustainable and resilient future. His research focuses on harmful algae blooms and their ecology, on nutrient cycling and aquatic ecosystems, watershed nutrient reduction strategies, and freshwater resource management. His research interests take him from small farm ponds to the Laurentian Great Lakes and involve analyzing data sets from a single Minnesota lake to thousands of lakes scattered across the continental United States. Before arriving in Duluth, Chris studied lake nutrient enrichment and toxic cyanobacteria blooms in one of the most intensively managed agricultural regions in the world as a research scientist at Iowa State University. He has an undergraduate degree in biology from the University of Texas at Austin and a PhD in biology from Baylor University. Well, Chris, it's over to you.
All right. Thank you, John, for the introduction. I will go ahead and get my slides set up here. Thank you very much, John, and thank you, everyone, for joining me today on one of these beautiful summer days we have here in northern Minnesota. I'm always happy to talk about harmful algal blooms, so I really do cherish this opportunity. I really don't need to set up an introduction after the introduction John gave. That was very nice, but I do always like to point out that I have been studying cyanobacteria for almost 2 decades now. I feel like I'm getting old. I originally started thinking about cyanobacteria in drinking water reservoirs of central Texas and how they impact drinking water and drinking water treatment approaches and costs of those. And like John said, after that, I moved up to Iowa, where I studied a huge population of these really intense agricultural lakes, which are dominated for a good portion of the time by cyanobacteria. Up here, I originally thought I was going to have to pivot to a new research focus when I got up here because of the nice pristine waters of northern Minnesota lakes that I heard about all my life. But then in 2018, we had this cyanobacteria bloom in Lake Superior when I was coming back on a transit from the Sioux Locks back to Duluth. And that really started increasing awareness of cyanobacteria blooms up here in the region and people reporting more and more cyanobacteria blooms. So it became kind of an emerging threat to the region. I work with communities a lot to help identify cyanobacteria. That's just an image of a lake sample we got from Cook County Soil and Water Conservation District that we looked at under a microscope to see if there were potentially toxic cyanobacteria present. And there were. So that's just a little bit more about my background. Before we kick into really kind of the practical nature of cyanobacteria blooms and how you can help keep your community safe and how you can help mitigate like the impacts of cyanobacteria, I just want to give a brief introduction on cyanobacteria and why you should care. So cyanobacteria are also known as blue-green algae, but they're not actually algae. They're not eukaryotic organisms. They are bacteria. So the interesting thing is we think about cyanobacteria like other types of algae, like green algae and diatoms, because they're within that community in the water and they undergo photosynthesis. So they're photoautotrophs and kind of form the base of those food webs. But microcystis is more related to E. coli, and algae is more related to things like green plants and trees, terrestrial green plants, than they are to each other, which is always really interesting to think about. They can take on a whole host of different morphologies, from little single-celled organisms to these gelatinous colonies like that image of microcystis you see there, and also some of the filamentous types of cyanobacteria, filamentous colonial ones, that form these big chains that are really diagnostic for trying to identify what taxonomic group they belong to. So I always like to show this slide because most people think about cyanobacteria. This is a new thing because I'm finally hearing about it and it's starting to impact some of the systems I'm familiar with. But the really interesting thing about cyanobacteria is they have been on Earth for a long time. They evolved about 3.2 billion years ago on Earth. So when we tend to think about cyanobacteria, we tend to think about them in the context of today's climate. But actually, cyanobacteria have evolved under a diverse set of different climates throughout Earth's history. So it's always interesting to think about their functional adaptations based on, when those could have been involved in the past and the reason potentially why those traits were developed. And so this is my Cindy Hagley from Minnesota Sea Grant, former Minnesota Sea Grant employee slide. I do want to make the case that cyanobacteria are natural members of lakes. And so seeing cyanobacteria in a lake isn't like a sign of impending doom, right? Cyanobacteria do a lot of good things. Because of cyanobacteria, we have oxygen in our atmosphere on Earth. So they were the original photosynthetic organisms that helped produce a lot of that oxygen that we have in our atmosphere. And they also form the base of the food web, like a lot of other photoautotrophs in the algal communities within lakes. So they help support the fish that you end up catching and eating and enjoying. So they are a good thing. So your question may be, well, why are we here then if they're such a good thing? Why are we talking today? And that's because in certain circumstances, cyanobacteria have a lot of adverse effects, both on public health and on the environment. The thing that I think most people are probably familiar with are that cyanobacteria, under certain circumstances, they can produce cyanobacterial toxins, which either attack the liver or attack the nervous system, or just give you like a irritation on your skin that's a little annoying and kind of itchy. Kind of the poster child for cyanobacteria was back in 2014, I think it was now. I forget the exact date. Sorry about that. But a cyanobacteria bloom in Lake Erie, the toxins ended up getting taken into the drinking water intake for the treatment plant, and they had to shut down the water supply for Toledo and the surrounding area for about a week. So these do have a significant impact. Besides toxins, they can also impact the environment. So cyanobacteria blooms if you look at that right picture there, that's an example of a cyanobacteria bloom. They rapidly form this huge, dense mat of organic matter. And as that organic matter decomposes, it consumes a lot of the oxygen in lakes that can result in fish kills. So it's a public health threat. It's also a threat to the habitat of lakes. And so here's an example. Minnesota Pollution Control has the motto, when in doubt, stay out. If you look at a lake and it kind of looks odd, it looks like pea soup green or there's a surface scum on it, the best approach is just to stay out of that. But certain recreational activities are more or less risky towards being exposed to those cyanobacteria blooms. And the big impacts that we tend to think about are on children recreating in those lakes. The toxins can make people sick, and also pets. Every year in Minnesota, there are confirmed reports of dogs that have died after swimming in these waters and then licking their fur to get the cyanobacteria cells off them, which have the toxins in them after they get out of the water. And I should say that the dog deaths and things like that are likely underreported just because there's not a lot of awareness out there. And the reporting structure is a little challenging in the state at this point, but it's getting much better. And I'll touch on that in a second. All right. So when we think about cyanobacteria blooms, they are anticipated to become more frequent, severe, and longer lasting in the future under different climate change consequences. And I think in addition to this, like I said earlier, we're also seeing cyanobacteria blooms where we haven't seen them in the past, or at least where the current generation doesn't really remember them occurring in the past. So cyanobacteria blooms occurring in Lake Superior is one example. We also see them in some of the colder water, low nutrient boundary water canoe area wilderness lakes as well, where they haven't been in the past. And here are just a couple examples of why we anticipate that cyanobacteria are going to increase under climate change. Lots of these are based on physiological adaptations that the cyanobacteria have that favor their growth in competitive outcomes under a changing climate. Some of those are nutrients, more nutrients in the system. Also, how the warming atmosphere and warming waters will also give them an advantage. And I'm not really going to touch on these today just due to the time we have together, but I'm happy to talk more about these if anyone's curious. Like I said, one of the primary drivers that we know that helps cyanobacteria blooms become established, and this is based on probably 40 years of research at this point, is that cyanobacteria tend to thrive when nutrients are higher or become more enriched in a system. And there's a word for this we call cultural eutrophication. That's just the nutrient enrichment of a body of water. But here's what it's look like. Here's what it looks like. So this is an image showing a Secchi disc at 8 inches depth in a lake. And so you can see here we have a pretty low nutrient system. The waters are relatively clear. You can see that Secchi disc, that disc there in the water perfectly. But as lakes become more and more enriched, again, this is a Secchi disc at 8 inches depth, it becomes harder to see that as we lose water transparency due to increased nutrients. And along with that reduction in water clarity, we also see the community shifting from algae like diatoms and green algae and things like that over towards systems being dominated by cyanobacteria like the globular microcystin that you can see there. So it's this combination really of land use and climate that is driving the expansion and the increase in severity of these harmful algal blooms. And I do want to point out that nutrients can come from a lot of different sources in these bodies of water. There's external sources such as agricultural runoff, residential runoff, urban sources, but there's also atmospheric deposition of nutrients as well. And potentially these are contributing to some of the blooms that we're seeing in our more remote lakes that don't have that direct watershed impact. As waters warm up, they become more strongly stratified, meaning that they don't mix from top to bottom. And cyanobacteria gain an advantage under these conditions because they have certain mechanisms where they can float to the surface and shade out all other types of competitors below them. And the other aspect of climate change that we tend to think a lot about is the increase in extreme weather events, extreme precipitation events, but also the strengthening of droughts. So when we have stronger storms, we get higher flow coming into systems that can really load up the lake with a lot of nutrients. And if that's then followed by a relatively calm period with warmer days and not much flow, that's when we can really see these cyanobacteria start taking off in lakes and blooming. So our current challenge, though, is like we have 5 decades of research, right, on what causes cyanobacteria to develop. But now we're seeing them in these lakes where we shouldn't see them. And it's causing us to think a little bit about, well, what are the exact drivers of cyanobacteria blooms in these new systems? Because once we can identify those mechanisms controlling them, we can start thinking about, well, what are the solutions towards mitigating them? But we're having to go back to the drawing board a little bit to rethink about what these primary mechanisms are, which is just, to me, it's a really fascinating time to be studying cyanobacteria because it's a bit of a shift in like the paradigms that we've learned throughout our career. So to get to the practical side of things, I want to start with just talking about how you can help identify cyanobacteria blooms to keep yourselves, your family, your loved ones, and your neighbors and communities safe. And so I'm just going to start out with a slide showing the signs of a cyanobacteria bloom. And these are all images of a cyanobacteria bloom occurring on different lakes. But if you see water that looks pea soup green, like your old Campbell's pea soup that you used to have with a ham sandwich or something like that, that is likely the sign of a cyanobacteria bloom because that's the type of organism that tends to bloom in freshwater systems. If you see a nice surface scum on the surface of a lake that looks maybe like spilled paint or have streaks to it, that is also a sign. And lots of times you can pick up like this brilliant turquoise color as well, which is kind of another diagnostic characteristic of a bloom basically decaying. And then there's also floating globs or mats. So these are all telltale signs of what a cyanobacteria bloom looks like. But beware, there are imposters that every year I get calls about these other phenomena that can occur in lakes that are mistaken for cyanobacteria blooms. One is that lots of times you can get these big globs of filamentous green algae. That's the image on the left. And during the day, they can float to the surface as they begin to photosynthesize and produce oxygen that makes them buoyant. So that's often mistaken for a cyanobacteria bloom. The other one is plant pollen. Up here, we deal with a lot of plant pollen, and it kind of gives this yellowish sheen to a lake. So if you're reporting a cyanobacteria bloom back when we see a lot of pollen occurring, that's probably going to be my number one question to you is, have you noticed any pollen on your car lately? And if the answer is yes, it's likely not a cyanobacteria bloom. And over on the right, the other one is duckweed. I think most people are familiar with pondweed or duckweed. It's a little plant that tends to float on the surface of these water bodies. But again, that is not a type of cyanobacteria. And I do want to give a shout out. I hosted a workshop with the Lake Superior National Estuarine Research Reserve on cyanobacteria blooms about a month ago now, and some of these slides are from that presentation, so full credit to the LS-NER for that. So there's a couple simple tests that you can actually conduct at home to get a better handle on if something is a cyanobacteria bloom or not. One is basically the jar test. So take an old mason jar, take an old mayonnaise jar, take a water bottle, take whatever you have handy, collect a sample of that water, shake it up, and then just let it sit in the corner for like a couple hours to a day and see if the greenness in there floats to the surface. Like I said earlier, cyanobacteria can control their buoyancy in water and they can float to the surface. So if you do this and you have something float to the surface, it's likely a type of cyanobacteria, whereas if everything just sinks, it's likely another type of algae. For the filamentous green algae, you can also do the stick test. So if you, as biologists, we love to use sticks to like poke things, is like I always say. So if you take this stick and you poke it into the water and you can lift up these thread-like hairs of algae, that's likely a type of green algae. If you put your stick in the water and it really doesn't pick up anything on it, it just disperses in the water around it, then it is likely some type of cyanobacteria. And so a good point to make here, though, is, you know, we can look at things and see if something is a type of cyanobacteria or not, and we can identify it to a genus or species of cyanobacteria that can potentially produce toxins. But you don't actually know whether or not a bloom is toxic just by looking at it. So even though a certain strain of cyanobacteria can produce toxins, doesn't mean they're producing toxins at that moment. The other challenge is that there's a lot of cyanobacteria that look very similar. They're exactly the same. They're different strains of the same species. And one species may have the genetic machinery or the capabilities to produce toxins, but the other doesn't. And in the right image here, this is an image of Delicospermum lemermanii from Lake Superior. This is the primary bloom-forming organism that we've been seeing in Lake Superior. And the question was, oh my gosh, is this a potentially toxic strain? And due to the work from Cody Sheik at Large Lakes Observatory and others, we know that that strain of Delicospermum does not have the genes required to produce microcystins or antitoxins or saxitoxins. So that's actually a good thing. All right. And I also just want to point out that cyanobacteria blooms are very dynamic. The big satellite image is just an image of Lake Erie, again, kind of the poster child for thinking about cyanobacteria blooms. But you can see how that greenness is very spatially unevenly distributed throughout that lake. In the little inset image, I want to point out the fact that these cyanobacteria tend to move around lakes quite a bit. So often you'll have cyanobacteria float to the surface, and on a day with a gentle breeze, that cyanobacteria will be pushed with the waves to the downwind shoreline of that lake, where it accumulates and people notice that it is a cyanobacteria bloom. But as the winds can pick up later in the day and start mixing things, or the winds shift into a different direction, that bloom may be in one location, but then move to the exact opposite side of the lake later in the day. So these things are really dynamic, and it makes it hard to know whether or not toxins are present in the water just by looking at a bloom, because a bloom may have been on one shoreline in the morning, forming lots of those toxins and things like that. But then the greenness shifts to the other side of the lake, but the toxins remain in place. So again, we just have to be really vigilant when we think about these blooms and whether or not they're toxic. All right. And this was work done by my graduate student, Leah Schleppenbach, with funding from Minnesota Sea Grant. Oh my gosh, I can't even remember. It's been a while now. It's been about 4 years, I think. But we studied 6 lakes throughout the state of Minnesota. We had 2 lakes down near the cities. We had 2 lakes, Portage and Hill, kind of in central Minnesota. And then we were up on White Iron Lake and Tate Lake up in northeastern Minnesota. And one of the interesting things that came out of this project is Leah was able to find detectable microcystins in all of these lakes. Even the lakes that at the time didn't really look bloomy, there were background levels of microcystins within those lakes. I do want to point out, though, that all these lakes except Lake Pelcher were below the drinking water standard of 1 nanogram per milliliter. And only Pelcher Lake was above that and had certain occurrences where it was above the recreational limit of 6 nanograms per milliliter or 6 micrograms per liter. So it was just really surprising to kind of see these background levels where the system didn't look bloomy. The other thing I want to point out is we tend to use microcystin as an indicator toxin, saying if microcystin's there, there's likely other types of toxins there. But that's not always the case. So this is from the same project, and this is looking at microcystins, but also 2 other types of toxins, anatoxin A and cylindrospermopsin. Those are the different dashed lines, and these are neurotoxins, which can have huge impacts on the nervous system. And there were times where in lakes we saw the anatoxin and cylindrospermopsin levels higher than the microcystin levels. Again, so we need to take kind of this holistic approach about looking at multiple toxins when we're thinking about toxicity and health risk of these systems. I do want to point out before I move, like when we measure these things in the lab, we use a lot of fancy instruments. This is one of the few pictures of me in the laboratory because I don't get in there much anymore. But this is an instrument we got that basically automates the analysis of cyanotoxins for our lab. It's basically like a little robot that goes back and forth and pipette samples and reagents. And then we set up the instrument, we leave, and we come back and we have results. So that's really nice. But I do want to point out that you don't need fancy instruments to measure toxins. And I think this is kind of a new development. Sorry, I have that little bar I'm just noticing. I hope that hasn't been in your way the whole time. But there are kits that you can purchase to kind of get a rough estimate of whether or not toxins are present in your water. And you can actually get like a qualitative estimate of what that range may be. This was research done by Janae Whitaker over at the Lake Superior National Estuarine Research Reserve. There's a QR code there that you can snap, and Janae basically looked at 3 different types of cyanobacteria test kits that people or local organizations, lake associations, soil and water conservation districts can purchase. And she has a full report on that, but the ones offered by Gold Standard Diagnostics were kind of the ones that had the best accuracy and gave you the most information. I should say I tend to think about these as like a screening approach or technique, right? So if you get a hit of cyanobacterial toxins, microcystin in this case, to actually get a nice quantitative accurate estimate of what those concentrations are, you would likely need to send that sample to a laboratory for further testing. All right. And now the practical side of this whole talk. So how can you help protect yourself and others? And basically, how can you contribute to, trying to solve this problem of cyanobacteria blooms in these natural resources that we just love so much? And the first one, I'm going to put this slide up there. The first is if you suspect any kind of illness from harmful algal blooms, if urgent, call 911. If it's your pet, call your veterinarian right away. I've been on a lot of message boards now, and there is a lot of great information that veterinarians have to help diagnose these things, even better than probably a decade ago. So they can really help out with your pets. If it's not urgent, like an urgent health related issue, you can report this illness to the Minnesota Department of Health Waterborne Diseases Unit at the numbers listed here. And the other thing I want to point out is that Minnesota Department of Health is now recommending that you can also report harmful algal blooms and track them using the US EPA's Bloom Watch app. So this is just a little app you can get in the App Store, download it to your phone. And the reason I want to bring this up is because if you're at a body of water, you can look on this app to see whether or not someone has tried to report a bloom recently to see if there's a history of bloom activity in that lake. Um, and the other thing is, the information provided in the app is only going to be as good as the user community that supports it. So if we can adopt more and more users using this, I think that'll help more communities out going forward and help people keep safe and make good decisions when they're choosing where to recreate on lakes. Okay, so on top of that, there's a lot of different approaches that have been put out there to solve this challenge of harmful algal blooms or mitigate the consequences. I gotta say, some of the things are very maybe outlandish, maybe a little sci-fi, and the efficacy of those approaches may not be all that great, but there's stuff you can go on Amazon or you can go on the web and you can buy all kinds of stuff to try to solve these challenges. But there are some tried and true approaches, and I'll talk a little bit more about those in detail. I think the one thing to point out is that some of these methods and these management approaches are geared towards different audiences, right? There's things you can do at your home to help prevent these things. There's things communities can do to help prevent harmful algal blooms or mitigate their impacts on kind of a larger scale on like public bodies of water. And so it's just really important to think about that when you're thinking about some of these different treatment approaches. And for the sake of today's talk, I'm really going to focus on what you can do at your home to help mitigate kind of the occurrence of these cyanobacteria blooms. And the first one, like I said earlier, the correlation between harmful algal blooms and nutrients, I think, is still a pretty tight one in most systems. So one of the things you can do to help reduce harmful algal blooms is basically by reducing nutrient inputs from your yard. So when I talk about this, I'm talking about fertilizing only when needed. If you don't know that you need to fertilize, you can always send a soil test to the University of Minnesota extension offices, and they will test it, I believe, for free. Don't send me the bill if that's not the case, but I believe you can do that. And then you can see what you actually need to fertilize with. One of the things is phosphorus tends to be the limiting nutrient in freshwater ecosystems. So using a phosphorus-free fertilizer is always recommended, and in some states it is mandated. Mostly your lawn needs. If you are a shoreline owner on a lake, don't fertilize up to the shoreline, have a certain buffer or setback there, trying to prevent grass clippings and all the nutrients bound up in that grass from entering waterways would be a good thing. And picking up your pet waste. I am the happy owner of two beagles that mostly drive me crazy most of the time. But there's a study that has been conducted. I think John Downing was part of it back in the day, but lots of waste and pet waste and things like that, and sewage is really high in phosphorus content compared to other types of organic matter. So you get a huge load of phosphorus just from pet waste. So remember, there is no poop fairy. Pick up the poop, help do your part. And I mean, that's one of the joys of being a pet owner, right? Okay, sorry, I got to go back here. All right. The other way you can help is by capturing nutrients before they get to the water. And there are a lot of publications that can tell you about DIY, stormwater management approaches. Some of the ones that always come to mind for me are building rain barrels to, again, capture that water and slowly release it and reuse it. Rain gardens. Shoreline buffers are a really big approach. The image on the right shows like a shoreline buffer just going in versus one that has become established. So having those natural shorelines, and all of this is in an effort for those plants to help sequester or take up fine nutrients before they get into your water. And finally, properly maintaining septic systems is key. Faulty septic systems can result in the release of a lot of nutrients before it's actually being treated to our waterways. Get septic smart. Here's a QR tag to a nice publication you can look at. I got to admit, before I moved to Minnesota, I was never on a septic system in my life. So I had to learn a lot about septic system as I was getting a new septic system put into my house and how to properly maintain that. So there's a lot of great guidance out there for how you can contribute to that if you are on a septic system. And again, I just want to reiterate the point that stormwater management and kind of these approaches are a community effort. There's things you can do, there's things the county can do, the city can do, So you're not in it alone. It's a collective effort to help mitigate these impacts to really protect the cherished natural resources that we love to recreate at. So this is my final slide. Basically, cyanobacteria are this threat to both public health and habitat. They are both a legacy kind of issue and an emerging issue in new areas. And we really anticipate that they will get worse in the future under different aspects of climate change. Temperatures, one I talked about, extreme weather events, flood drought cycles, and the strengthening of those. All of these are going to have an impact. You can identify cyanobacteria blooms visually, but you really can't tell if they're toxic just by looking at it. So hopefully, I'm always available. If you have a sample, you want someone to look at it, my contact information is on the next slide. Feel free to reach out to me. But I hope what this does is this gives you a little bit of information where you can triage some of those situations that aren't necessarily cyanobacteria blooms and make those informed decisions before reaching out. We can help increase awareness by reporting these blooms, and we can also help reduce cyanobacteria risk by really managing nutrients and stormwater, and you can play a big part in that. I also want to point out that there are a lot of opportunities for training on cyanobacteria blooms and how you can keep people safe that are being offered. I mentioned the one we just had at the Lake Superior National Estuarine Research Reserve like a month ago. I think they're going to roll out another one of those in near Bayfield in the month or so. So that'll be another training session. If you're in Cook County on Monday, I'm going to be up in Cook County for a cHABs workshop talking about cHABs and lots of these similar issues and approaches that you can use. And also, Hillary Sorensen from Minnesota Sea Grant and I got funding. We finally got the funding approved from the state Environmental and Natural Resources Trust Fund last year, where we are going to be working with communities. We're going to be working with lake associations and soil and water conservation districts to provide people with tools to monitor and track both harmful algal blooms, so toxins present in the water using the test kits I talked about today, but also looking at chloride, which is another huge issue impacting our ecosystems. And with that, thank you all for taking the time out of your busy, gorgeous summer day. They're kind of dwindling as we move forward, as we're already in August. So thank you very much, and I would be happy to take any questions.
Thanks so much, Chris. That was fabulous. Really, really terrific. And thanks also for mentioning septic systems. If people want to learn more about septic systems, go to itascawaters.org and look at the Practical Water Wisdom June recording. Fantastic information on septics, and also for stormwater management. And Chris, I'm not sure you knew this, but the first Thursday in September, John Bellotta and Maggie Karshnia are giving a talk on how to manage stormwater on your land. So thanks for all those mentions. That was terrific. Now let's get to the questions. Handling this part of today's program is Bill Granches, manager of Itasca County's Aquatic Invasive Species Program and a member of Itasca Waters Board. Bill?
Thank you, John, and hello, everyone. Just a reminder, submit your questions using the Q&A function. We are not using a chat function. Now I'm going to read the questions aloud to Chris and then give him a chance to answer them. We may combine similar questions together. So if we don't get to your question and you'd still like an answer, just send us an email at info@itascawaters.org. And boy, the questions are just pouring in right now. Okay, first question for Chris. Surface water bodies that discharge to groundwater. Is there a risk of cyanobacteria or their toxins reaching the groundwater? If so, is there a general time frame for attenuation after which the risk becomes negligible? I'm particularly concerned about groundwater sources used for drinking water that have a direct hydraulic connection to surface water.
Yeah, thank you for the question. Cyanobacterial toxins can degrade in the environment, but they can also persist, right? So we can see them like persist through time. And I know there's been a couple of examples of people like finding measurable cyanobacterial toxins by looking at past sediment cores going back like decades to centuries. So they do persist. So I think it would be largely dependent on the, how much hydraulic exchange you have there between the groundwater and the surface water. To my knowledge, I have not seen this come up anywhere as an issue that people are concerned with, or, there's not routine testing of cyanobacterial toxins from groundwater. So I think it is a pretty minimal risk. But I think, it's really going to depend on what that connectivity looks like between the surface water and the groundwater.
Very good. Next question, Chris. Is the shorter ice season contributing to more severe harmful algal blooms in our region?
Yeah, that's another excellent observation. I think that's one of the aspects of climate change that we're really concerned about is, as we start losing ice cover in these winter months, basically, are we going to start shifting the whole seasonal progression of algal communities forward in time? So, like right now, up here at least, we tend to see these cyanobacteria blooms really kicking off in, September, October is when we tend to get them reported the most, except on like some extreme systems where they're, present year round. So the thought would be, if we get earlier ice out and things start warming up sooner, will those start pushing into like July and August and then June and July? And I think that's what we're concerned with, because then not only do you have them occurring earlier in the season, but you also have them lasting longer in that lake, and they can form all these positive feedback loops where they're just going to continue, being in this state of being cyanobacterial dominated. So yeah, it's not just the increased water surface temperature and stratification, but definitely that loss of ice is going to have an impact.
Absolutely. It's affecting aquatic invasive species too. Okay, next question. Have you done any screening for anotoxin?
Anatoxin is one that we have not gotten hits on for, at least down here. Lots of the research we're conducting right now is in nearshore Lake Superior and also the St. Louis River estuary. We've had hits of toxins in the St. Louis River estuary, but they've primarily been microcystins and then saxitoxin. We have not had a hit for anatoxin yet. But I know Cody Sheik for the Large Lakes Observatory has been doing some genetic profiling of cyanobacteria across Minnesota. And I believe he said that they had been seeing anatoxin genes in some of those cyanobacteria strains. So that's one of the ones in addition to kind of microcystin and saxitoxin that we're really looking into is anatoxin. Cylindrospermopsin is another one, but to my knowledge, there aren't a lot of hits on that yet. I think that's a lower threat than the other three.
Okay, very good. Next question. You emphasized phosphorus as a key trigger for the blooms, but what about other nutrients like nitrogen and carbon and other toxic substances such as iron and various heavy metals?
Yeah, I mean nitrogen plays a big role as well in kind of shifting the communities more or less towards one type of algae or cyanobacteria or another. So one of the things I really didn't go into is when systems tend to be scarce in nitrogen relative to phosphorus, cyanobacteria are expected to thrive because they can fix atmospheric nitrogen in these little cells called heterocytes. So when other organisms are limited due to low nitrogen, cyanobacteria aren't. They can persist and keep growing and have that competitive advantage. So I think one of the things is, phosphorus tends to limit these ecosystems, but most ecosystems are co-limited by both nitrogen and phosphorus. And lots of the approaches you take to reduce phosphorus also help with the reduction of nitrogen. So I think you kind of get a lot of times both nutrients. It's only like in the most extreme agricultural settings where you have these really high like nitrate and ammonia levels that you really target that nitrogen to help reduce, public health impacts just from high nitrogen itself.
I see. Okay. Next question. Do zebra mussels worsen harmful algal blooms by filtering out good algae and leaving behind cyanobacteria? Are we seeing this in Minnesota? And how does this affect cyanobacterial harmful algae blooms?
Yeah, that's really like one of the issues that we're starting to pay attention to more and more with aquatic invasive species, particularly mussels. There's been a lot of research on selective filter feeding of mussels. And I think, depending on the study, there's cases made supporting both sides of that. But there are reports where zebra mussels can selectively filter out things other than cyanobacteria. And so those cyanobacteria go back into the water column. I think the other point to make behind that as well is, as the mussels are going ahead and consuming those algae and those cyanobacteria, they're taking in that organic matter, but they're also excreting a lot of phosphorus, a lot of nitrogen, and a lot of the cyanobacterial toxins themselves and putting it into kind of that dissolved form in the water column. So I think there's a lot of feedbacks between aquatic invasive species and cyanobacteria. So remember that slide I had when I talked about like the combination of land use and climate and things like that. Definitely throw aquatic invasive species into that mix. And you can see why this is such a challenging issue to help resolve.
Absolutely. Next question. In your upcoming work with Hilary Swanson, will you be monitoring a chloride? Oh, sorry. Will you be monitoring for chloride as a potential driver of cyanobacteria, which are more tolerant than other algae, or for overall impacts of chloride?
Yep. For us, it's more thinking about what are some of the big threats to ecosystems and people. And the two that came out that you hear a lot about right now are chloride and cyanobacterial toxins, right? So we're going to be collecting data on both of those because depending on where a group is located or where a lake is located, one may be much more important than the other, right?
Sure.
So we want to give people the tools to be able to help with both of these challenges that we're facing here in Minnesota. Whether or not there's some kind of correlation between the two, that really wasn't the goal of this study. That being said, we'll have data on both of these things from lakes, so we could potentially look at that. But I don't think there's any strong evidence for chloride being a driver of harmful algal blooms at this point.
Okay. Well, speaking of challenges, is there any, is there any evidence that wildfire smoke through nutrient fallout or changes in light and temperature might influence the harmful algal blooms in our lakes?
Okay. So I swear I did not put this question in there myself. So one of the other things that my lab is doing a lot of research right now is how wildfires are impacting the water quality in our lakes. And from that perspective, we've seen that not smoke necessarily, but direct watershed burns tend to lead to more nutrients being loaded into a lake, more soils and sediment that make the water turbid. Again, that might give cyanobacteria an advantage because they can float to the surface. But one of the interesting things we saw in this population of lakes we've been looking at due to the Greenwood fire that happened back in 2021 is we didn't see a response of the overall algal biomass to those increased nutrients because they moved from nutrient limitation to light limitation in those systems.
Mm-hmm.
And we still haven't seen the recovery. But one of the things that I tell my lab about all the time is why was I not smart enough to add phytoplankton composition analysis to what we're doing? Because I think that could potentially be a driver. We just don't have the data on it, just simply due to the cost of the project.
Sure.
But wildfire smoke is the exact same thing. When you get fallout of smoke and ash and things like that, you get heavy metals, you get nutrients along with it. You also have less light getting to that system, so maybe a little bit of reduced primary productivity there. So I think we still don't know what those overall impacts of smoke are, but it's something I know that other researchers are actively looking into, and my lab is starting to look into it as well.
Fantastic. Thank you for that. Are there examples of successful programs in northern Minnesota or nearby states that have reduced harmful algae blooms, outbreaks?
Yeah, I'm trying to think of specific examples, and I don't really have any that come to mind right now. Lots of the ones I see that are really successful are kind of at a smaller scale. So like maybe someone's managing a pond on their property or something like that, where they can implement different approaches at like nutrient reduction or keeping the animals and waste out to help lessen those. I know in agricultural regions, there's a lot of straw barley additions that people use to release tannins into the water that are supposed to have an impact. You can use artificial mixing in some lakes. And this has actually been used in a lot of like stormwater ponds and smaller lakes where they put these little aerators that go throughout the system and just kind of churn up the water. Really, and that is meant to prevent cyanobacteria from floating to the surface and gaining that competitive advantage. One of the things you have to worry about is that you're not drawing up nutrients from the bottom water and just spraying them everywhere on the surface. But those are techniques. I think from the land management perspective, the buffer strips, the reduced nutrient loads, and all that, we know do make a significant impact on at least the nutrient loads going into the system, which should have a cascading effect on cyanobacteria.
Sure. Sure. Well, what steps are state and local agencies taking to reduce nutrient runoff into our lakes?
Yeah, I think a lot of the good work is really happening with like agricultural extension. I know they're working a lot in the agricultural regions of the state just to trying to minimize those impacts. Thinking about, retaining nutrients on the field and preventing it from running off into receiving waters. Soil and water conservation districts throughout the state are doing a lot of good work with people to think about like public set or setbacks on properties and buffer strips and things like that. I know there's a lot of conservation programs and easement programs that people can take advantage of to reduce nutrients. And then just all the good work I think that people are doing as parts of community because there is more awareness of just thinking about nutrient reduction on your lawn. I mean, on your actual lawn and then thinking about approaches for people managing stormwater. I know up here, just driving through Duluth, I mean, I pass by so many places that have rain barrels
Yep.
or these really pretty, like I remember when I was in Iowa, there was one lake we were studying, and the community built this just gorgeous rain garden that was very attractive, but also was really effective in helping reduce nutrient runoff from an area where they knew they had a problem because we helped
Mhm.
identify that. So there's a ton of great work out there being done by just a whole host of organizations. And just real quick, from like the Minnesota Department of Health perspective and the Minnesota Pollution Control Agency, they are really working on this challenge of cyanobacteria blooms at the state level. Again, part of the challenge is, we have limited funding resources that we have to target different systems with. So this is where you can really help out. You can help those agencies by helping inform them on where these problems are, where they're occurring, so they can target, response and funding to help mitigate those problems.
Very good. Well, I see we're running out of time here, Chris. Thank you so much. I'm going to turn this back over to you now, John.
Hey, thanks so much, Bill. Really, and Chris, thank you so much for the terrific presentation. I'd also wanted to mention that if you want to learn more about Itasca Waters and what the county program on AIS, you can find Bill Granchis at the Itasca County Fair coming up real soon now. So another opportunity. So thank you again, Chris, and thanks so much, Bill, for running the Q&A. This program will be available soon on our website, itascawaters.org. Please feel free to recommend it to family, friends, and neighbors. Also, we'll be asking you for your ideas about the Water Wisdom series, so you can use the survey link that you'll receive after this Zoom presentation, or see on the screen, or respond to the evaluation form. We'll email you. The next program will be on Thursday, September 4th, and feature Maggie Karshnia and John Bellotta from the University of Minnesota Water Resources Center and Minnesota Sea Grant will provide practical solutions to help property owners prevent, minimize, and manage stormwater runoff. Also, Chris mentioned wildfires. There'll be a presentation by Sarah Elliott in the same series on Thursday, October 2nd, where she will talk about wildfires and water quality. You can register for free on itascawaters.org for any of these webinars coming up. I'm John Downing, and thanks so much for watching today.